Hey, it's Mr. YS, and this is the final clip for 1.6 on cyclins, the cell cycle, and the causes of cancer. So here we can see a chart, and we're looking at the different stages of the cell cycle. So we've got section M, which M is for mitosis, and we've looked at the stages of mitosis, which were PMAT, prophase, metaphase, anaphase, and telophase. And we also had a look at these other sections, G1, S, and G2, all of which make up interphase. So a cell's always between divisions. It's either in interphase, where it might spend about 90% of its time, or it's in mitosis and it's dividing. So we've got these proteins, and they're called cyclins. And we'll get to why they're called cyclins, and it's to do with the fact that they cycle in and out in terms of their concentration. So they're not like other proteins in that they're there for a period and then they disappear. And it's because they're a signal for the cell. So the cyclins increase in concentration and then decrease. And so we can see that they're associated with different parts, and they're associated with checkpoints. If they don't rise high enough, then we can't go to the next stage. So if a cell has just divided, if it's just done mitosis, then in order for it to progress through phase G1, where it might be copying organelles, then we need to get a rise in cyclin D. Now, what these individual cyclins are is not important. You just need to know overall what they do. But we can see here cyclin D rises during G1. And that rise to a high concentration, this is what creates a kind of checkpoint. It will only rise to this high concentration if everything is okay in the cell. And then we'll progress to the next one. And so we get cyclin E rises. So the first one was D, cyclin E rises during S phase, so DNA replication. And if everything's gone right with the cell, we'll also get cyclin A rising, and then we'll get the next stage and we'll get cyclin A rising, and so on. So these proteins, they rise in concentration and they act like a checkpoint. If things are not okay, then we will get the cell exiting the cell cycle. It will exit the cell cycle, and it might even, in a very noble way, kill itself, cell apoptosis. So it might decide if there is, for instance, in S phase, if there is DNA damage detected, then through other mechanisms it can be detected. Things will bind to DNA damage, and they will lead to the cell stopping there. It wouldn't progress into G2. It's going to exit. It might try and repair the damage, but if it can't, it would then, if everything's going right, it would kill itself. The cell would release digestive enzymes from the lysosome and kill itself. So what does a cyclin actually do? So we said that they increased in concentration. Let's just have a look at what they do. And this isn't, you don't need to know this in any big detail. But here we have a cyclin. And what it does is it binds to something called a CDK, a cyclin dependent kinase. It depends on the cyclin to be activated. So if the cyclin raises in concentration, and if the cyclin can then bind to the cyclin-dependent kinase, the kinase then becomes activated. So this is now active. And what it will do is it will go and it will add this phosphate group. So reminder of what a phosphate group is. It'll go and to these proteins, so target 1, 2, and 3, they're all proteins, and quite often they're enzymes, and now these will become activated. So it's kind of an activation pathway. If we have cyclin, it will bind to CDKs, cyclin-dependent kinases. It will then, the cyclin-dependent kinase, it will add these phosphate groups on, so it's phosphorylation. And now we get active enzymes, and that will cause the next phase, or the phase that it's in, to continue. And what happens if we don't have cyclin is it remains inactive. This cyclin-dependent kinase isn't active. We don't get phosphorylation. And so we then, if this was, for instance, in S phase, we wouldn't get all the DNA replication being switched on. So what's the role of cyclin? Here's our summary. Cyclins are proteins that help to control the cell cycle. They make sure that the right tasks are carried out at the right time. So they bind to CDKs, cyclin-dependent kinases, to activate them, and then they can start attaching phosphate groups to other proteins, for instance enzymes, and activating them. And that will cause the cell cycle to proceed. And we've got different cyclins triggering G1, S, G2, and then finally triggering mitosis. Here's a bit of nature of science which has been included. This here is a sea urchin, and in a case of lucky coincidence, which is called serendipity. In a case of serendipity, a researcher who was looking at sea urchins and actually looking at nothing to do with mitosis discovered the presence of cyclins. And so the way that this was found was they were tracking through radioactivity. They were tracking proteins, and they were running them out, and this is a kind of, it's almost like chromatography. They were running out proteins, and they were looking at when those proteins were present. So they were doing it after time intervals. And when they looked at the proteins in different time intervals, normally what you'd expect, it was in a fertilized cell of the urchin. And so as it kind of grows and develops, you'd expect the proteins to get higher in concentration. So if this was protein concentration, you'd expect the concentration to just keep going up and for these bands to get darker. And that's what they found for most things. As time went on, there was an increase in the darkness of the bands. But they found something strange, which was that for a couple of the proteins which had been separated, they didn't see that. They saw that it was not there, and then strongly there, and then not there, and then strongly there. It was cycling in and out. And that was really strange, because that's not what the other proteins were doing. So they saw these two cycling in and out. And so here the M is for mitosis. This isn't exactly what they got. This is a kind of summary. And this is for interphase. And they found that these two different proteins, one of them was cycling in and out during the period between mitosis in interphase, and one of them was only becoming increased during mitosis. And this was this is explained by the fact that they have a specific role in the cell cycle, and they're only there when they need to be there. So this one was increasing during mitosis. The other one was needed for during interphase. Here's our quick summary. So cyclins relied on serendipity, lucky chance. Researching sea urchins, it was hunt, although I doubt they'll ask you that. And they found that particular proteins increased in concentration after fertilization and then decreased. But they'd expect all of them to just keep increasing in concentration. So they found that it rapidly increased and decreased, and this was at different phases, either during mitosis or in interphase. And so they named them cyclins because they were cycling in and out, although he didn't set out to discover cell cycle regulation. How does cancer form? Well, here we've got the two scenarios. One, we've got normal cells. And for normal cells, if they don't have growth factors, things which are causing their growth, then there is no division. So they're not always dividing. Their division is dependent on their environment, and they're receiving signals, and that leads to them either dividing or not. They could be dividing to replace worn out cells, and these cells as well could be stem cells, which are going to divide. And the stem cell will create one cell which will start to differentiate into different types of tissue, and one cell will remain as a stem cell. So in some cases, with growth factors, they will divide, but it's a controlled division. Whereas in cancer cells, we can see that whether they have growth factors or not, they still divide. It's continuous. It's now not responding to signals from elsewhere, and we can say it's uncontrolled. And so a tumour is the growth of these cells in an uncontrolled manner. And the other thing about cancer cells is that they don't perform their usual function, because something's gone wrong, or lots of things have gone wrong in the cell. It's no longer carrying out the function of that tissue. So as it grows and expands into the tissue, it's not something that's useful. So what happens in normal cells? Well, in normal cells, if there is some kind of damage, e.g. there's some DNA damage, and maybe it's from a mutagen, a chemical or radiation that causes mutations. If there's DNA damage, then it will be sensed. There's some proteins which will, in the case of DNA damage, these proteins bind to the damage and they lead to signals, and those signals cause the cell to enter cell death, which is called apoptosis. And so that's a good thing. So any cell that has damage, it shouldn't reproduce, it shouldn't go through the cell cycle, it should stop, and if it can't fix it, it should kill itself. But what happens with a cancer cell is that it gets DNA damage, and maybe it's had mutations in some of these proteins that should be binding to the DNA damage and sending a signal. And so instead of it being able to respond as it should do, it now doesn't have that signal, the message to say that there's damage, and it doesn't enter into apoptosis, and it just keeps dividing again and again and again. Okay, this bit to do with P53 is a little bit beyond IB, but I think it kind of explains what's happening, especially for those people who are interested in a little bit more to do with cancer genetics. So there's one protein which is implicated in a lot of cancers, and it's called P53. What P53 does is where there's DNA damage, P53 goes and it binds to the DNA where it's damaged. And what this does is it causes a halt at the G1 checkpoint. It would try and activate the repair, but if it doesn't work, it then triggers cell death. So it's kind of like it's one of the heroes of the cell in that it's trying to detect damage and trying to stop the cell from going any further. But if we then don't have normal P53, if we get mutations of this gene for the protein, if we get lots of mutations in this gene, then it's going to remove that. And so instead of halting at the G1 checkpoint, it would progress. So here's our first way that we could cause cancer. We could have mutations in genes that are meant to stop cancer. These are called tumour suppressors, although that's not really part of the IB syllabus. So we can mutate, if we mutate genes which are supposed to stop cancer, because they're involved in the cell cycle, then that could be one way that we could lead to cancer. So the other way that we could get cancer being caused is where we get mutations in things which are called oncogenes. And so oncogenes are genes where if we have a mutation, some kind of mutation, it could lead to, for instance, an increase in the amount that this gene is used. It could be some kind of growth factor, some kind of protein that is involved in increasing cell division. So if we mutate it and we cause an increase, then we would get more of that growth factor, leading to more cell division. So in a way, oncogenes are like the accelerator, because we're kind of causing something to increase through mutations. One of the ways this can happen is due to smoking and other things. You can get chromosomes where one bit gets chopped off and it might get added on to the end of another chromosome. So we might get this kind of joining. And now, because it's in the wrong place, it might be next to something that leads to increased expression. So there are lots of ways it can happen, but that's one of them. So if we get increases in the expression of certain genes, they can cause more cell division, and that's an oncogene. Now, often in tumours, it's mutations in both oncogenes and tumour suppressors. So a tumour suppressor is like the brakes. So if you affect the brakes of a car and the accelerator, then you're going to get this increased cell growth and division, so uncontrolled cell division. So here's a kind of hypothetical series of events. So what could happen is, initially, you get some kind of mutation that switches off a regulator in the cell cycle that's a negative. It might be helping to kind of reduce cell division. Next, you get a different mutation somewhere else that overactivates a positive cell cycle regulator. So you've removed the brakes and now you're stepping on the accelerator. A third mutation might inactivate something which is supposed to be helping to keep your DNA the same. And then we can get lots of mutations rapidly accumulating until now, after many mutations, it's a cancer cell. Its brakes aren't working. The genes that make it want to divide have been increased, or their expression's been increased through mutation, and it's no longer able to keep its genome the same, keep the DNA the same. And so this is what creates a cancer cell. Here's our summary. So what is cancer and what are tumours? Tumours are abnormal groups of cells with uncontrolled cell division. They divide continuously. Now there's two different types. There's malignant, where they can detach and move and grow in different locations. And then there are other ones which are called benign, where they do stick to each other. They don't invade other tissues. So if you're getting tested for cancer, it's great news if you hear that your tumour, whatever growth it was benign. This means it's not going to spread. So cancer is caused by mutations to DNA, to genes, and that means a change in the base sequence, change in the sequence of nucleotides. and it's often caused by mutagens, but it can just be errors in replication, errors in copying the DNA. And in particular, if we get changes to oncogenes, they are genes capable of leading to cancer. These we talked about being kind of like the accelerator, because they're going to be involved in cell cycle regulation or promoting cell division. And often it's several mutations which are necessary, but it could be due to carcinogens, e.g. cigarette smoke, radiation, pollution, and UV light. One of the best ways that you can cause mutations is by smoking. So smoking contains thousands of chemicals, and lots of them we now know are mutagens. Or we could use the term carcinogenic, meaning that they cause mutations in DNA. And so for a while, the cigarette companies were saying that their products didn't cause cancer. And we can see here cigarette smoke per day, and we can see the occurrence of cancer. And we can see it's a very strong positive correlation. But they were arguing that causation hadn't been shown. That although there was a correlation, maybe it was other things that were causing it. So now we know, and we've tested it, we've extracted the chemicals, we know the chemicals, and we can show that those chemicals, they lead to tumours. And this has been done in lots of animal studies as well. Here we go. So this is way beyond IB, but this is just one thing to show you what's happening with cigarette smoke. So cigarette smoke contains all of these chemicals, and what they do is they go in, and you can see, I'm just going to highlight this here, they bind to DNA. And you can see it's making this covalent bond, in the case here, to an adenine, to that base. Well, actually, it's to the nucleotide. So you can see it's bound there. Now, what then happens is that it sticks where it is, and it causes a replication error. It causes, you can see there's an issue there. We've got an A to A. And what then happens is, well, your repair machinery come along and try and cut this out. It gets cut out, but now it pairs back. Now we've got T to A. So we've gone from what was an A to T has gone to T to A, just through smoking these chemicals, which then bind to your DNA. So as your DNA tries to repair, you get these mutations in. Now all of this is, you don't need to really know it, but I just want you to see the mechanism. So mutagens can bind to DNA, causing these mutations to happen. And here's our summary. So there's a very, very strong correlation between smoking and cancer. As you increase smoking rates, so does the risk of developing cancer. And there's a huge increase in death rate due to cancers of the esophagus, pharynx, larynx, and lungs in smokers versus non-smokers. But death rate due to other cancers, where it's not, the smoke isn't going over and touching the tissue, is not that different. Smoking does actually increase the rate of some other non-respiratory system cancers as well. But the argument was it was not causation. But now we have the evidence. We know how these carcinogenic compounds cause mutations, and so now there's no doubt that smoking causes cancer.